for giving me the opportunity. This result is actually part of my PhD project, and I have nothing to disclose. Purinergic P2X7 receptor plays an important role in inflammation. By formation of a large non-selective membrane pore, it increases cell permeability, results in apoptosis and cell death. In our previous study, we have confirmed the dense expression of P2X7 receptor on urothelial layer of porcine bladder. We also have shown that P2X7 may be involved in acrolein-induced urothelial damage. Acrolein, as we know, is a highly toxic urinary metabolite of cyclophosphamide, and it can cause severe bladder cystitis in chemo patients. In this study, using transepithelial electrical resistance or TEER value, which is a well-known indicator of epithelial monolayer integrity, we have explored the effect of acrolein on the urothelial barrier integrity and the role of P2X7 receptor in this effect. We have also aimed to determine whether P2X7 receptor dependent large pore formation is associated with urothelial cell damage using UPRO1 uptake assay, which I will explain in details later. As a result, TEER was stable for controls. Acrolein caused 60% reduction in TEER values for up to 48 hours. However, pre-incubation with P2X7 selective antagonist has significantly protected the cells from acrolein-induced TEER reduction. ZO1 tight junction protein immunocytochemistry has confirmed this result. With higher magnification, we can clearly see the damaging effect of acrolein on ZO1, which is significantly protected by P2X7 selective antagonist. In UPRO1 uptake assay, high concentration of ATP or BZ-ATP, which is a selective P2X7 agonist, was exposed to cells. The UPRO1 is a large molecule that is only able to pass through this large pore, binds to nucleic acids, and generates a fluorescent signal. To investigate the role of P2X7 receptor, two selective antagonists were applied to urothelial cells. So primary urothelial cells treated with different concentrations of ATP and BZ-ATP showed a concentration-dependent increase in fluorescence intensity, indicative of pore formation. However, both antagonists significantly reduced the pore formation activity of BZ-ATP. Also, comparable results has achieved using ATP, which is not shown here. As a summary, acrolein causes damage to urothelial barrier integrity, which was protected by P2X7 antagonist. Also, P2X7 activation plays a major role in urothelial pore formation. In conclusion, P2X7 receptor plays an important role in bladder inflammation, and inhibition of this receptor could be a pathway to treat bladder cystitis. Thank you.
Thank you very much. Any questions from the floor? Okay. Mike?
You're not thinking it, Mike Ruggieri from Philadelphia, sorry. It's not the P2X, it's actually a ligand gated ion channel.
Yeah.
So it's not that pore you're talking about. It's the pore that's created by the acrolein itself, which is not the same as the P2X receptor pore.
Okay, you mean like the link between these two?
How does this work?
Yeah, well, we think the link is ATP release. That's one from one side. So if initial damage to the urothelial cells by acrolein can cause ATP release, which we know a concentration of ATP intracellular is usually much more higher than extracellular spaces, whereas there are studies that shown in terms of inflammation or cystitis, we have this ATP release. So if acrolein is causing enormous amounts of ATP release to extracellular spaces, and then that ATP is actually activating P2X7 receptor on underlying layers and kind of started a downstream signaling in those layers. So we kind of protecting the cells from those ATP stimulated signaling. The other link can be apoptosis. So we know acrolein is causing apoptosis by actually a variety of different mechanisms. It can be direct and indirect mechanism, of course. So indirect mechanism, it causes DNA and protein adduction, whereas in indirect mechanism, it can cause stress like mitochondrial stress, ER stress, and oxidative stress, which we think is very dose dependent. So now we think in a lower doses, which is the point that it causes oxidative stress or ER stress, that's when P2X7 can have a role, and then P2X7 antagonism is blocking apoptosis in this part, which we know P2X7 is very famous with causing apoptosis, and antagonists can block the apoptosis. Actually, in this setup with tier value, we tried higher dose of acrolein, which is 100 micromolar, and in that higher dose, we couldn't get the protective effect of the antagonist. Yeah, sorry.
So this would be that the higher dose of acrolein was starting to damage the more cells, the urothelial cells closer to the lamina propria, and not just the umbrella cells with a lower dose.
By releasing the ATP release from those urothelial cells, that like from the damage of the damaged cells, there are enormous amounts of ATP release probably that can actually activate the P2X7 with the other cells.
Okay, thank you. Very nice study.
Thank you.
Okay.
Yeah, well, I have one question. You're
Sure.
very careful in your conclusion, and you don't conclude more than you can conclude from your basic results. You say that this is relevant for acrolein cystitis, induced cystitis, and you can find an acrolein-induced cystitis also in humans. But cyclophosphamide-induced cystitis is also used as a model of interstitial cystitis. Do you think that it is a model of interstitial cystitis?
It's really hard to say in terms of, because definition actually is changing every year, I guess, and it's hard to say because it's more called hemorrhagic cystitis, and some people believe that it's different from interstitial cystitis. But if we have inflammation in common in these conditions, then P2X7 antagonist can block inflammation. That it can be relevant, but yeah, I'm not sure if this model is actually a model of interstitial cystitis.
But think of it. You instill a toxic substance into the bladder.
Yeah.
In interstitial cystitis, nobody has isolated a toxic substance that induces inflammation.
No.
So it's not. I just want to stress this, that
Sure.
what you have seen is not a model of interstitial cystitis. It's a model of acrolein-induced cystitis, and that is very clinically relevant.
Yeah.
Thank you.
Definitely. Thanks for your suggestion.
Okay. Thank you again. And